Hi Year 12s. Today we're going to be looking at cell division. These videos are coming before cell metabolism, which is our next topic, and I'll talk about the reasons for that in class. But for now we're going to look at cell division. There's going to be two videos for this PowerPoint. The first one that we're looking at now is going to be talking about binary fission and mitosis, and the second video will be alone just looking at meiosis. So let's get started. So this is your need to know that you're going to need to be making note of. So as per usual, these are all taken from the Stage 2 summary document on the SACE website, and these are all the dot points that we'll be addressing in these two videos. I'm not going to go through them. If you want to take note of them, pause the video here and make notes for yourself. So duplication of DNA. So duplication of DNA is referring to DNA replication. So DNA replication is always the first process that needs to occur before cell division takes place. So when DNA replication takes place, every DNA molecule and chromosome inside the cell basically makes a copy of itself. At this point, they become visible as chromatids, and the two chromatids are joined together by a centromere. It's important to note that while the number of chromosomes is staying the same, so for example, when DNA replication happens inside our bodies, inside our cells still only have 46 chromosomes. But the amount of DNA that's there is doubled. So the chromosome number doesn't double, just the amount of DNA that's there. I've given a picture on the right here, which we'll put this fella down. Basically, you've got your two chromatids, and the whole thing is considered to be a chromosome. The type of DNA replication that we actually do in our bodies is known as semi-conservative, and we can see a semi-conservative DNA replication happening down here. The reason that we know that DNA replicates in a semi-conservative way is due to the way that the structure of DNA was deciphered by Watson and Crick in 1953, which basically suggested that each strand of the double helix of DNA would serve as a template for a synthesis for a new strand. So we've got a double strand, each one of those strands, better in this diagram here than my hands actually, each one of these strands here would serve as the template for a new strand of DNA. So that when a new molecule of DNA is produced, it's made up of one old strand, which is the blue, and one new strand, which is the red. So it's semi-conservative because we've got one old strand and one new strand of DNA. And the daughter molecules there are made up of one old, one new. So, why have we talked about DNA replication? Because no matter which type of cell division we're talking about, DNA replication has to take place first. Now, binary fission and mitosis we're going to talk about. They're very similar to what we've covered in Stage 1 biology, if you've done Stage 1 biology. So some of this information may be repeat, but it's important you know it again because it does appear in your Stage 2 summaries. So, as we discussed before, binary fission is just basically meaning division in half. The word binary fission just translates to division in half. Most bacteria, which undergo binary fission, contain only a single circular chromosome that contains several hundreds of genes. So it doesn't have much DNA that needs to replicate, or that we need to be making sure is copied in each new cell. That chromosome does still contain double-stranded DNA, and like I said before, it has to replicate before our DNA or our binary fission can actually take place. Binary fission generally can go between 20 minutes to several hours, depending on how many resources are available to that bacteria when they're going through binary fission. And binary fission occurs in 4 stages, or 4 steps, but it's only really one process known as binary fission. So the first step is that DNA replication occurs, and it doubles the genetic information in the singular chromosome. Still only one chromosome, but double the genetic information. That original chromosome and the duplicated chromosome, which are still attached together, attach to the cell membrane, and the entire cell begins to elongate. So we can see that here. So doesn't show it's attached, but the chromosome here is attached to the cell membrane. As the cell starts to elongate, those chromosomes are physically separated by the fact that the cell membrane is moving as it elongates. Eventually, the cell grows so large that the cell membrane starts to invaginate, and then it physically separates the cytoplasm and the cell, creating 2 copies of the cell, each with their own copy of the chromosome. Mitosis. So that's binary fission. That's all we're going to look at binary fission. Now we're going to look at mitosis. So First off, mitosis is a lot more complicated than binary fission. And that's because binary fission is occurring in prokaryotic cells, where mitosis is occurring in eukaryotic cells. Why does it need to be more complicated? Well, first off, eukaryotes have organelles, prokaryotes don't. So we don't need to worry about coding for those in binary fission. Eukaryotes, we need to worry about coding for those organelles to make sure the organelles are all recreated in the new cell. Eukaryotes have 2 or more linear chromosomes. So for humans, for example, we have 46 chromosomes. Prokaryotes have 1 circular chromosome. Okay, so we have to worry about duplicating and copying and making sure we've got in both cells 46 chromosomes. Binary fission has to deal with 1. It's more complicated to deal with 46 than it is to deal with 1. And lastly, human chromosomes metachromatin, which is a mixture of DNA and protein. So it's not just the DNA they're dealing with there. Mitosis itself. So the mitosis that we're going to talk about occurs in 2 different stages, mitosis and cytokinesis. Both of those have their own functions. Mitosis itself is the division of the parent nuclei into 2 daughter nuclei, while cytokinesis is the division of the parent cell into 2 daughter cells. The genetic information in the daughter cells is kept exactly the same through physical separation of the sister chromatids into single-stranded chromatids, which we'll talk about a little bit more in the coming slides. The steps of mitosis: prophase, metaphase, anaphase, telophase, cytokinesis. Let's start talking about those. So we'll start with prophase. So during prophase, the chromosomes start to condense, which means they coil up. And because of this, we can now view them under a light microscope. So we can see this stage under a light microscope. The nuclear membrane also starts to break down, as we can see by the dotted orange lines around here. It's starting to break up. And that allows the chromosomes to be released into the cytoplasm of the cell. We also have our centrioles, which are cylindrical structures of protein, move to opposite poles of the cells. So we have our centrioles, which are moving to opposite poles of the cells, and those centrioles are also starting to produce microtubules called spindle fibers. And those spindle fibers will be really important in the next stage. So in metaphase, our spindle fibers that we've got coming out of our centrosomes are going to attach to the centromere. And the centromere, remembering, is the point at which 2 sister chromatids are joined. Once they're attached to those centromeres, the spindle fibers begin to contract, and they have equal force pulling on them. So as they have equal force pulling from each pole, the sister chromatids are forced into the equator of the cell. So over here, because of the force being pulled on them, they're all pulled into the equator of the cell. Now this could also be called the metaphase plate. In anaphase, the spindle fibers continue to pull on those sister chromatids. Now because they've already moved to the center of the cell, the equator of the cell, there's nowhere more that these sister chromatids can go. So when we're continuing to apply force to them, what happens is the centromeres break, and the resulting sister chromatids, now separated, can actually move to opposite sides of the cell. So we're putting so much force on that those centromeres are breaking, sister chromatids being pulled to opposite sides of the cell. Once that occurs, the parent cell begins to elongate, and it's also important for a future stage that proteins, actin and myosin, start to assemble at the equator of the parent cell during this stage. Telophase. So during telophase, the nuclear membranes are beginning to form again, which we can see through these yellowish orange dots here. So we're starting to form again. And they're forming around the 2 sets of chromosomes which we've just separated. That creates a cell nucleus at each pole, one of which belongs to each of the soon-to-be created cells. The chromosomes become less condensed, so we're no longer able to see them under a light microscope. And it's also at this point that we end the stage of mitosis. So the division of the nucleus now is finished. We've started with 1 nuclei, we've ended with 2 exact copies of the same nuclei. All that's left is cytokinesis. So in cytokinesis, an indentation begins to form in the cytoplasm of the cell. So this is our indentation here. In animal cells, we call this a cleavage furrow. Now, in cytokinesis, the proteins that we talked about in anaphase, actin and myosin, they start to form what's known as a contractile ring around the center of the cell. Now, that contractile ring, as the name suggests, starts to contract or condense, which pinches on the cell and increases the size of that cleavage furrow. So it's increasing it, making it more indented. As they continue to contract or condense, they continue to do so until the cell physically can't be pinched anymore and it breaks into two daughter cells. And that's the cell division stage. So the cleavage furrow and the contractile ring are what's physically ripping those cells in half. It's important to know that all cells that are produced via mitosis are pretty much identical copies of the parent cell, but that's only assuming there's been no mutations that occurred in DNA replication. If there's mutations that have occurred in the DNA replication, of course there may be some minor differences in the cells. The cells will always have the same number of chromosomes, and they'll be the same type of chromosomes. And mitosis is also going to ensure that always the cells that are produced through mitosis are classified as diploid, which we'll talk about a little bit more in the next video. You need to be able to compare the two processes of binary fission and mitosis. So first off, similarities. Both processes, chromosomes are separated only after DNA is replicated. Both the types of cell division that can be used for growth or reproduction, both of which are asexual reproduction, not sexual, and both processes also include one parent cell being split into two daughter cells. The differences, on the other hand, are that binary fission only occurs in prokaryotic cells, whereas mitosis will only occur in eukaryotic cells. Prokaryotics are going to divide in one stage made up of 4 steps. Eukaryotes divide into 2 stages, which is mitosis, which has 4 steps, and then cytokinesis. Chromosomes are separated by the cell membrane in binary fission, whereas in mitosis they're separated by the spindle fibers. And again, mitosis is a more complex process which has more highly organized steps.